Hello, everyone. Welcome to today's talk by Dr. Niklas Bray. I can say doctor because he just finished his thesis in August. He was working with me in my lab. Niklas actually had a very wild journey through his research. So he started, he did his, he graduated, he did his BSc degree in 2018, but during his BSc degree, he joined my lab as an undergraduate. And at the time, I had some data from the Derek Bowie lab. He recorded neural excitability in Purkinje cells. And I thought at the time that Niklas can work on that data. They were interested in understanding the role of NaV 1.5 channels that apparently are expressed in these neurons. And typically, these neurons are expressed in the heart. But that was a novel finding, is that they're expressed also in the brain. So we thought, let's look at that data and work on it. But then we discovered that the project was going nowhere. We need to actually understand the kinetics of NaV 1.5. So he actually focused on that data, on NaV 1.5 channels in isolation and see what the kinetics are. So he started his PhD work in September 2018, I believe, as a PhD QLS student. So he fast-tracked into the PhD and continued working on it. And then he published a paper as well on the topic. But during his work, another collaborator of mine approached me, Gilles Blourde, and told us that he has this EEG data that he wanted us to work on and see what are underlying the neural rhythms within that data. So Niklas decided to work on it as well. And since then, he didn't look back. He actually went on and made quite a number of discoveries in that project. And I believe today's talk is going to be mostly focused on this. So I look forward to hearing it, and good luck.
Thank you. As Anwar said, I'm a recent PhD graduate, and I'd like to talk to you about the part of my thesis related to modeling and understanding aperiodic EEG signals. So if the brain is thought of as an orchestra, then brain's thought of as an orchestra, then these are the players. This is an image of a top-down view of a mouse brain, about 1 millimeter squared. And here are a bunch of neurons that are expressing a protein that fluoresces, that glows when the membrane potential of these cells changes. And if you look at the changes in the electric membrane potential of these cells over time, you would see something like this. You can see that there's these spikes, and these spikes reflect the output of these neurons called action potential. If you look at this data, you'll notice that these spikes like to cluster together. They like to happen at the same time. And these bouts of action potentials recur at a certain, say, around 100 milliseconds or so. This phenomenon is referred to as synchronous neural oscillations. Synchronous because this spiking tends to happen synchronously, and oscillations because these bouts tend to recur rhythmically. And these sorts of neural oscillations have been observed in many different brain areas at many different frequencies and have been associated with different brain functions. In fact, there's become a taxonomy to describe these neural oscillations, and we named them by Greek letters. And here's just a table of some of the well-known brain rhythms. You can see in the right-hand column, these are the sorts of brain states they're associated with, ranging from alpha rhythms all the way to theta rhythms. For example, when you're all sleeping, your brain likes to operate by firing about once every second synchronously. Now, you might know how I could possibly know what your brains are doing while you're sleeping, because you're not going to let me put a microscope through your skull and image. And surprisingly, when these neurons all fire synchronously at the same time, they generate such large electric fields that you can actually measure these signals from outside the skull. And this is a technique called the electroencephalogram, where you glue electrodes to the scalp, and then you can see the changes in the electric field that you can record. And this technique is relatively simple. In fact, it's been done for 100 years. It was in 1924 that the very first EEG signals were recorded by this guy called Hans Berger. And he saw a rhythmic fluctuation at about 10 hertz, which is called now the alpha rhythm. Alpha because it was the first rhythm to be discovered, which is why in the previous chart, the Greek letters are completely all over the place because they're named in the sequence that they were discovered. But if you look at an EEG, you don't see a nice, beautiful sinusoid that's fluctuating at whatever brain rhythm you're looking at. EEG signals look a bit more like this. In fact, this is an actual EEG signal, and you might be able to make out these slow fluctuations in the signal. And if you zoom in a bit further, you might see these faster oscillations in the signal. But clearly just visual inspection of the time series doesn't lend itself to identifying brain rhythms, especially ones that are very small. And a way to solve this problem is by using a mathematical transformation called the Fourier transform, where you can decompose the signal into the weighted sum of many different frequency components. For example, this signal you can see is described as many sinusoids at different frequencies and different amplitudes. And if you plot the amplitude of every wave as a function of its frequency, you get what's called the power spectrum. And what the spectrum indicates is the relative contribution of each of these frequencies to the EEG signal. In this perspective, you can see very clearly these two peaks, which reflect these particularly strong contributions of frequencies to the signal. And these reflect about 0.5 and 8 hertz. These reflect the delta rhythm and the alpha rhythm I was just describing. What's abundantly clear in this alternate view of the EEG is that there's a lot more signal that doesn't lie in these peaks. This signal is sometimes referred to as aperiodic EEG. It's sometimes referred as the spectral trend because this is a trend in the power spectrum. And this is the part of the signal that I'm going to be focusing on today. This has become particularly interesting in the last couple of years because about 6 years ago, it was suggested that whereas most people have taken this part of the data, treated it as noise and just tried to ignore it or remove it, recently people have suggested that it should be analyzed very explicitly in its own right. This is a paper that came out in Nature Neuroscience where they argued that the overall spectral trend follows a very straight line in log space, and that there's two implications of this. First of all, compare the left-hand side and the right-hand side, you can see that this alpha peak here has disappeared. And their argument is that whereas most people would say that the amplitude of this alpha oscillation has decreased from up here to down here, they would argue that there's actually zero alpha power here. And so you should be measuring the amplitude of these brain rhythms relative to this background spectral trend, also known as detrending the signal of this aperiodic component. The other implication of this finding is that the aperiodic exponent or slope of this line is an interesting feature in its own right, should be extracted and analyzed, and it tells you something completely independent from the brain rhythms. In fact, since this paper was published, there's been an exponential increase in the number of studies using this method to study aperiodic EEG. They've been correlating changes in the slope to many things from different brain states to different diseases. And for just a couple of examples, it's been associated with sleep. Depending on what stage of sleep you're in, the EEG power spectrum seems to go steeper and steeper. When you give different drugs to individuals, such as ketamine, propofol, or xenon, you also see changes in the slope of this EEG spectrum. And it's also been associated with aging. So while all these changes in the spectral slope suggest that it's not just noise, that it has something to do with the brain, we don't really know what this means. So the outstanding questions in the field, despite the fact that this method has been applied many times, is what is the physiological mechanism that produces the spectral trend? Is the spectral slope actually meaningful? Just because we can correlate it to things, does it mean something physiologically? And do EEG spectra need to be trended? Is it okay just measuring the absolute amplitude of these brain rhythms? If it's not, that suggests that a lot of the EEG analysis done in the past 100 years might be wrong. If detrending is necessary, how should you do it? This is the power spectrum of the sea level in Bermuda. It shows these two strong peaks, which reflects the periodic tide as the moon orbits the Earth. You also see that there's this overall spectral trend, which in this case is caused by aperiodic fluctuations in sea level as the wind speed and atmospheric pressure change. On the other hand, here is the power spectrum. Here's the spectrum of the emissions from a star in the Taurus constellation. It also exhibits an overall spectral trend. And there's also these three peaks here. Now, in this case, the trend reflects the colors of light that are emitted by any object in the universe that's at a certain temperature. You'll notice that if something glows red hot, This is the distribution of colors that it actually emits. Now, in this case, these dips are caused by the absorption of light at certain frequencies, in this case by hydrogen ions. And so the amplitude of this dip reflects absorption. And so the amplitude makes sense to be defined relative to what would otherwise be there. On the other hand, if the wind stops blowing in Bermuda, the moon's not going to stop orbiting the Earth. And so unlike the right case, the aperiodic, the broadband component, the spectral trend in this signal, and the periodic components really have nothing to do with each other. All that to say, there are mechanistic explanations to be found for the spectral trends observed throughout nature. Just because there is a spectral trend doesn't mean that it should be detrended. And also understanding how the peaks and the spectral trend relate to each other may be accomplished by understanding the actual mechanisms that produce the signal. So what are the theories for how the spectral trend is generated? Well, there's two main theories and they contradict each other, maybe not surprisingly. The first theory is that the EEG spectral trend doesn't really exist. That's just an epiphenomenon of brain rhythms and everything is a brain rhythm. Under this theory, the brain exhibits many different oscillations. This is a compendium of all the oscillations that have been discovered in the brain, from very slow rhythms or delta rhythms around 1 Hertz, the faster gamma oscillations. And it's been argued in the early 2000s that these slower rhythms span over larger parts of the brain, whereas the faster gamma oscillations are only very local in the brain. And the consequence of this is slower oscillations produce rhythms that are larger, faster oscillations produce rhythms that are slower. And so you should expect an overall trend to appear. And if you're taking the signal, you're taking the power spectrum, these peaks kind of blur out into what looks like kind of a trend, but it's not really. So that's theory one. And under this regime, no detrending is necessary. All the power reflects brain rhythms, so you can just look at the power, and that's brain rhythm. Then the alternate theory is that this spectral trend reflects something distinct, reflects some non-oscillatory activity, and that EEG spectra require detrending if you want to properly analyze brain rhythms. Now, I'm going to go into a bit of the background for this because some of the mathematical framework will then be extended in some of the modeling that I'm going to show. But the most basic model of non-oscillatory activity in the brain is the Poisson process. And basically what a Poisson process is just a random sequence of events. You can't really get more random than this. And this is a very often used model of brain activity. If you record from a single neuron in the brain, its firing looks quite a lot like this, which you might find surprising, but there are actually many large literature why you might expect deterministic neural systems to give rise to Poissonian-like spiking. Now, the EEG signal is typically thought to reflect synaptic currents, the currents that are caused when one neuron communicates with another neuron. And so with these synaptic timing events in place, each one is going to cause some sort of current and will produce something that looks like this, which is essentially every single time you have an event, you have some increase in the current and then it slowly decays. Under this model where the EEG signal reflects all the synaptic currents happening in the brain, you can just take many of these time series, add them up, get a pseudo model EEG signal, and you can show purely theoretically, in fact, that the power spectrum of this signal is going to follow something that looks like this. As you can see, it's not flat, it already shows some sort of trend to it, and the power spectrum is completely determined by this function. This is the frequency, and this constant tau is the decay time of these currents.
I'm just thinking that if you have a sum of many Poisson distributions, I think you just get a Poisson distribution
Yes.
with a higher frequency.
Yes, exactly. Yeah, that's true. So as I said, this is, I mean, it's hard to argue against the mathematics of this, but one of the reasons why neural oscillations are thought to be the bedrock of EEG is because, as I mentioned, when they're oscillating synchronously, they generate such large electric fields that they can be detected. And it's not entirely clear that Poisson spiking in the brain could ever generate detectable EEG signals. So the very first question I asked is it even possible for this sort of activity to generate EEG signals, even if it does have the right sort of spectral shape? And to answer this, I took that sort of framework of aperiodic EEG and then used very rigorous biophysical modeling to try to estimate EEG signals. In other words, given this sort of activity, of neurons, how does this turn into that, this EEG signal? If you were to take one of these neurons, which is a top-down view, and turn it sideways, this is what the neuron would look like. So at the center here, this is the soma, and this is the compartment that initiates this output signal, this action potential I was talking about. And then all around it, there's these dendrites, which is the sort of tree of neural processes, and they're like antennae that receive all the communication from other neurons. These are the locations where these synaptic currents occur. And what we did is we simulated these neurons receiving, getting bombarded with all this synaptic activity, and simulated the electric fields they would generate. And this is what one of these simulations look like. You can see these fluctuations in the electric field as the synapses come. That's the flash of an action potential. And you can notice as you just look at this simulation that most of the fluctuations in the field aren't caused by the actual outputs of the neurons. They're caused by the synaptic currents, the inputs to these neurons. So this gives us the local electric field. We're interested in the EEG signal. So what we can do is we can take this model, we can localize it somewhere in a head model, which is essentially a model of all the different conductivities of the neural tissue of the scalp or of the skull. And we can translate the current sources generated by this model into the electric potential we'd see at this electrode on the scalp. And this gives us essentially the EEG signal you would get from a single neuron. You can see here these two spikes, which are the action potentials I was talking about. And then there's all the sub-threshold fluctuations, which is caused by all the synaptic input. Now you might wonder how simulating single neuron EEGs could possibly help us understand the ensemble EEG, which is generated by something like 16 billion neurons. And there's actually a very clear mathematical framework why this might help us. And this was the core of our hypothesis at the beginning of this study, which is that due to the superposition principle of electric fields, an electric field is just superposition of all the electric fields that generate it. So we can decompose the EEG signal as the sum of all the contributions from individual neurons. Now, if you take the power spectrum of both sides, you would get that the power spectrum of the EEG are these individual contributions plus this sort of cross term here. Okay. So here, PI is the power spectrum of the EEG from neuron I multiplied by neuron J. And this is scaled by this component here, which is the coherence. The coherence is essentially the correlation between the two neurons' EEG signals as a function of frequency. And almost all the modeling work done on EEG has focused on understanding coherence. It's thought that the reason you get an alpha rhythm is because neural activity is coherent at 10 hertz, which picks out a peak at 10 hertz. But look at where the single neuron EEG shows up in this equation. And you can see that shows up in two places. First of all, it shows up here as a factor that's added to the second component. So changes in these single neuron EEGs might have an additive effect to these spectral peaks. It's also multiplied with this coherence, so changes in the single neuron EEG could also multiply the spectral peaks. And these single neuron EEGs have actually never been characterized before. So our hypothesis was maybe by understanding these contributions, we could understand the spectral trend a bit better.
Well, I don't understand why you get the cross term. Is this
Sorry, the power
obvious?
spectrum
Am I
is,
missing something obvious?
you're missing the thing I didn't explain, which is that the power spectrum is the absolute magnitude squared of the Fourier transform. So once you take the square of this, you get the cross terms.
Okay, thank you.
So in order to understand single neuron EEG, we basically just threw a computer at it and looked at many different biophysical models of EEG generation. For example, we have these models, as I showed before, that are receiving all the synaptic input, and we can change many different parameters like the morphology of the neuron to capture the different types of neurons in the brain, the excitatory and inhibitory neurons, as well as the ones in the different layers of cortex, layer 1 through layer 6. And then there's all these biophysical parameters that change the electric properties of the neurons, and I'll describe some of these parameters as I go through my presentation. What we can do is we generate one of these models, we put it somewhere in the cortex, we generate a single neuron EEG signal, we calculate the power spectrum, and then we can do that while we twiddle all the dials and knobs on our simulations. And you can see that as we change all these parameters, the amplitude and the shape of these EEG spectra change, and we can look at how all the different parameters end up shaping the EEG spectrum. Now, building on the non-oscillatory activity that I mentioned before, all the activity coming into these neurons is just Poissonian activity. It's random, it's white noise, has a flat power spectrum. You can already see that the power spectrum of these single neuron EEG signals isn't flat. In fact, what these simulations showed is that the generation of EEG signals by a neuron can basically be described as a double low-pass filter. Here is the average spectrum, and you can see that can be described as this sort of double low-pass filter where we have this low-frequency cutoff that's related to the deactivation kinetics of GABA receptors, and this high-frequency cutoff, which is related to the deactivation kinetics of AMPA receptors. Now, AMPA receptors and GABA receptors are two proteins that mediate most synaptic currents in the brain. The excitatory synaptic current through AMPA receptors, the inhibitory synaptic currents through GABA receptors. Notably, what these simulations suggest is between 1 and 40 hertz, where the majority of EEG analysis is performed, it's really these GABA receptors that are most important in shaping these EEG spectra. And so this is something we wanted to test. And so we teamed up with this anesthesiologist at the Montreal Neurological Institute, who was recording the EEG signals of his patients as they underwent general anesthesia from a drug called propofol. And the mechanism of action for propofol is that it slows the deactivation kinetics of GABA receptors, increasing the amount of inhibition in the brain, leading to loss of consciousness. So we wanted to analyze the spectra of his patients as they lost consciousness, as they received this infusion of propofol. This is what some of the data looks like. And our model suggests that you should have this low frequency filtering due to the kinetics of GABA receptors. And so we try to fit this component to the power spectrum with the time constant, this frequency cutoff, which gives us around 16 milliseconds, which is within the physiological range of GABA receptor kinetics. And our expectation would be that, the rest of this spectrum can be explained by these couple of peaks. There seems to be a beta and alpha and this low frequency component to the signal, in addition to this sort of trend here. And our expectation is that after giving the patients propofol, this time constant should increase. And that's exactly what we see following propofol. You see there's this shift, this increase in the slope, which is characterized by this increase in this time constant. And what we can do is we can do this at every single time point during the infusion of propofol and build up a pseudo dose response curve, where on the x-axis here we have the estimated concentration of propofol. On the y-axis we have how much this time constant changes. And you can see that as propofol increases, the time constant in the EEG spectrum changes. Now you could also estimate this dose response curve empirically using slices of brain, throwing propofol on it, recording from the neurons and seeing how their inhibitory synaptic kinetics change. And when you do that, you get these data points from all these in vitro studies, and you can see that it lines up fairly well, suggesting that the changes we see in the power spectrum are quantitatively and qualitatively consistent with changes in GABA receptor kinetics. That's one insight from these single neuron EEG simulations, that low frequency EEG seems to be strongly shaped by GABA receptor kinetics. The other insight is, unlike the toy model where we just have the synaptic currents from the Poisson process, here you notice that we have real units on the y-axis here. So the second thing we can ask is, what is the amplitude with which a single neuron contributes to the EEG signal? And if we can compare this to data, you see that a single neuron contributes very, very little to the EEG signal. Now, under the regime that I've been modeling so far, where all the activity is just Poisson activity, it's just random and uncorrelated, the EEG signal from 16 billion neurons will just be this multiplied by 16 billion. And so when you do this, we can ask how large the EEG signal will be from a completely asynchronous cortex all firing together. And this tells us that it's still much too small to generate detectable EEG signals. And that's perhaps not too surprising, because despite the fact that individual neurons can be modeled with Poisson processes, we know that neural activity in the brain is correlated with each other. And these correlations will further boost the power that you see in this signal. So what this simulation tells us, maybe more importantly, is that the approximate 5 orders of magnitude by which we're off here must be due to correlations that we didn't consider yet in our modeling. And so this allows us to ask the deeper question, which is, what sort of neural activity can generate the requisite correlations to produce detectable EEG signals? Does activity need to be rhythmic in order to produce signals that are detectable, or can aperiodic activity also generate enough correlations to be detectable? thus far, I've just focused on two components to EEG. The contributions, the biophysics with which individual neurons generate electric fields, and then these forward models of EEG that map this to realistic amplitudes of EEG signals. But now I introduce the final part of the system of EEG, which is the network dynamics, which so far I've been assuming is completely asynchronous. In particular, what I wanted to ask is, the thing that I hypothesized is that in the brain, neurons aren't completely isolated from one another. They have these connections and these recurrent connections. And it was my hypothesis that the recurrent connections in the brain might produce a type of aperiodic activity that generates enough correlations for EEG. So what I did is I modeled a network of neurons where every single neuron here is either firing or not firing. And when it fires, there's a probability that it causes its neighbors to fire. And when you model one of these networks and simulate it, this is what you would get as the average firing rate of the network. And you can see that every now and then there's these excursions, these large increases in the firing rate of the network. And these are sometimes called neural avalanches. And the presence or absence of these neural avalanches depend on how you parameterize this model. In particular, the dynamics of this network is governed by something called the branching number. If this branching number is 0, then every spike that occurs in this neuron won't cause any other spikes, will cause 0 spikes in the network. And so this is the asynchronous activity that I was talking about before, where all the neurons are basically independent of one another. They're just firing by themselves. As you increase this branching number, you can say, well, what if every spike causes 0.5 spikes to occur in the network on average? In this case, you start to have more correlations in the activity as there's this causal propagation of spikes throughout the network. As you increase this branching number even further to say 0.98, now you can see these large cascades of neural activity, these avalanches I was talking about before, as these spikes propagate throughout the network. So you might wonder, well, what does the cortex actually look like? And it has been, this is a very interesting model because for many decades, it's been argued that the brain sits at basically m equals 1. If m is greater than 1, then every spike causes more than 1 spike on average, and the entire network will explode exponentially. So m equals 1 is a critical state of the network. And it's been argued that the brain sits right at criticality, because it's been argued that at criticality, there are certain benefits and information processing, et cetera, that it might take advantage of. A couple of years ago, however, it was argued that, well, all the neural recordings we have of the brain severely undersample the brain. If this is what the brain looks like, then we can maybe record every couple of neurons. Okay? And they showed that if you're severely undersampling the brain like this, basically you're missing all these neurons here, half the neurons, 90% of the neurons. And so you're not going to actually estimate the branching number of the network accurately. They developed a statistical method that they argue estimates it much more accurately based on this undersampling. You can see here, this is the sort of conventional approach for estimating the branching number of neural networks. This is their estimate. As at full sampling, if you sample all the neurons in the brain, it does very well. But as you sample fewer and fewer of the neurons, the previous estimates for the branching number are wrong. Theirs is very similar. So all that to say, using some new statistical methods, you can put an electrode in the brain, see how the spiking happens, and then try to estimate this branching number of cortical circuits. When they do that, they find surprisingly consistent results when they put these electrodes in monkeys, cats, and rats, that the brain activity seems to obey the branching numbers that are not 1, but not 0 either. That hovers around, say, 0.99, 0.98, which is a slightly subcritical regime. So this gives us a plausible region for this parameter value. And going back to our model, we wanted to know how large the EEG signals could become. Can this sort of network activity generate sufficient correlations to generate detectable EEG signals? So what we did is we took these neural networks, we took 2 of the neurons in the networks and did our full biophysical simulations and asked how correlated the electric fields would be between these 2 neurons. These 2 neurons embedded in the network, how correlated can their electric fields be? You can see that this is the synaptic input into these two neurons. You can see these avalanches I'm talking about coming into the neurons, these clusters of synaptic activity bombarding them. And the single neuron EEGs, you can see, are quite correlated. Now we can do this simulation for many different networks with different branching numbers. And you can see that as the branching number goes to 0, the correlations die out because everything is completely uncorrelated. As the branching numbers come to 1, the activity in the network is sufficiently synchronous to drive correlated electric fields. And if we then ask how much these correlations should boost the overall EEG signal, we can see that as the branching number approaches 1, we can generate quite large electric fields, much higher than what you would get in a completely asynchronous case. So not only could the intrinsic synchrony that's brought about by these propagations of spikes through cortical circuits actually produce aperiodic EEG signals that would be detectable, you'd also note that the spectrum changes a bit. At these lower branching values, this is just the scaling that you got from the synaptic kinetics. And as you increase the branching number, you also add this additional power at these lower frequencies. So maybe not only can the propagation of spikes through cortical circuits explain the amplitude of the spectral trend, but it also captures this lower frequency component that can't be explained by synaptic kinetics by themselves. So broadly, this result demonstrates that EEG need not be exclusively rhythmic, that the natural aperiodic activity in the brain could generate detectable aperiodic EEG signals. All that to say, both this modeling as well as the propofol experiments suggest that not everything about the EEG reflects brain rhythms. At the very least, changes in GABA receptor kinetics can lead to changes in the EEG signal that doesn't reflect brain rhythms. So even if this theory is maybe non-oscillatory activity or non-oscillatory properties of the brain do contribute to EEG signals, what does this mean for actually analyzing EEG signals? Do EEG spectra require detrending? So the answer is it depends. You can imagine two scenarios. In one case, you have a brain, and maybe there's these two parts of the brain that are mostly independent of one another. One is oscillating, and the other has this aperiodic activity I was just talking about, in which case the EEG signal is going to be related to both of these dynamics. And if you were to say increase the aperiodic component, maybe just increase the firing rate of these neurons in the aperiodic population, you would increase the spectral slope, but because you're not touching the oscillatory component, this peak is barely affected at all. In this case, if you were to define the amplitude of this peak relative to the spectral trend, you'd say that this rhythm has decreased in amplitude, even though we know that it hasn't changed. On the other hand, if you were to say change these GABA receptor kinetics I was talking about before, you're changing the shape of the currents, the synaptic currents that generate the EEG signal. And if you do this, then you're basically changing the filtering applied to all activity coming out of the brain, and you increase the aperiodic component and you increase the oscillatory component equally. So in this case, the amplitude of these peaks relative to the spectral trend is maintained. And this is true also for the aperiodic component. You're changing the filtering that's applied to the brain. And so this is multiplicatively scaling everything. All that to say, if the point of EEG is to understand the dynamics of neural activity, you're trying to understand something about the timing of these synaptic inputs, whether they're oscillating, what frequency they're oscillating at. And if we want to understand these neural dynamics, then things that change this need to be corrected for, because this is changing how the EEG signal is produced, and this is going to produce changes in your results regardless of what's changing in the dynamics. So I suggested one thing, these GABA receptor kinetics that can change these synaptic currents. What else can? Well, basically any biophysical parameter in neurons can change this output. This is just a table of some of them. This is the GABA receptor kinetics. You can change the leak currents. You can change the conductance of these synaptic currents. And each one is going to produce its own impact on how EEG signals are produced. And they're going to produce EEG signal changes that have nothing to do with the neural dynamics. That's just one example here. Let's say that I've increased the conductance of leak channels in the neuron. You can see that if you don't account for this change in the spectral slope, you're going to say that between these two conditions, you have changes in delta rhythms, theta rhythms, alpha rhythms, decreasing gamma oscillations, which is not true. So if you were to account for this change in the spectral trend by, say, fitting it, and then detrending the signal, and then comparing the power after detrending, you would then get a much more accurate picture of the dynamics changing in the brain, which is that I ran these simulations, I know nothing changed, and that's better reflected after detrending the spectrum. So this is the theory. What does this mean in practice? Well, we have a great case study because, as I mentioned, our collaborator was collecting EEG data from subjects receiving propofol, and we'd expect that to be changing the spectrum in a way that would corrupt typical brain oscillation quantification. So we went back to this data. This is what it looked like at baseline. This is what it looks like after propofol. We can fit the changes in the power spectrum that can be explained by propofol's action on GABA receptors, and we can correct for it. And now we can compare the changes in the spectrum after accounting for these changes in synaptic kinetics. Now, people have looked at what propofol does to brain oscillations previously, and they typically find that there's an increase in delta power and an increase in alpha power that's associated with loss of consciousness. And if we were to do this analysis and look at changes in alpha, delta, alpha, and beta power over time without correcting anything, this is the result we get, which is fairly consistent with what people have seen in the past. You have an increase in delta and alpha power, you have a transient increase in beta, and then a decrease again. If we then reanalyze this data after correcting our spectra, we see that things look a bit different. Maybe the beta and the alpha power isn't so different, but then in the delta power, the dynamics change quite a lot. Basically, you have all this low frequency power that's now explained by changes in synaptic kinetics, not changes in rhythms. And this reveals what seems to be a change in delta rhythms that's much more time locked to when the patient's lost consciousness, whereas these alpha rhythms are not really affected at all by the detrending. So by detrending our spectra, it suggests that what used to look like a kind of lockstep increase in alpha and delta power is actually better reflected as an increase in alpha and beta power after the infusion of propofol, and then an increase in delta power after that, closer to when the patient's lost consciousness, suggesting that alpha and beta power are related to propofol infusion, but maybe it's only delta power that's actually related to its mechanism as a general anesthetic. So this is just one example of how correcting for the spectral trend might change your conclusions about what brain rhythms are changing. Now in part 3, I wanted to ask, well, everything I told you about so far has happened at quite low frequencies, between 1 and 40 hertz. What about the very fast EEG signals that are extending much higher? There's oscillations that have been detected in epilepsy as high as 600 hertz. What does the EEG spectral trend look like up there? Well, if you look at an EEG spectrum, it looks kind of like this. You can see that here you have this decreasing trend, and then it just kind of plateaus. So this might suggest that there is some other component that's adding to the spectral trend at these higher frequencies. But actually, if you take an individual, you inject them with a muscle paralytic, so they're completely paralyzed, and then record the EEG signal. And yeah, you can get ethics approval for this. This is what you see. So basically, EEG signals are extremely corrupted by muscle activity. All this signal that's up these higher frequencies is due to the electrical activity generated by muscles, not the electrical activity generated by the brain. So if you were to then analyze these EEG signals that are, well, the muscle activity has been attenuated, you can see that even up to these higher frequencies, the trend is basically explained by synaptic kinetics. In other words, the EEG spectral trend seems to be explained by synaptic kinetics as before, as well as muscle noise, and of course, amplifier noise, which is basically the internal fluctuations in the amplifiers. You can't record signals below that. So this suggests that there aren't any other mechanisms that actually explain the EEG spectral trend other than synaptic kinetics. The implication of this is that because of the filtering of synaptic kinetics, if you're to do something silly and just say, well, this is the amplitude of an alpha rhythm, if you're to have the exact same oscillation, but it's occurring at 250 hertz, well, then the filtering of the synaptic kinetics would mean that you only get this much power on the EEG signal. So the same amplitude oscillation in the brain will produce very different amplitude EEG oscillations. And in fact, you can see that if the oscillation is too fast, you're not going to be able to detect it at all on the EEG signal, simply because of how synaptic kinetics attenuate EEG signals. So one might rightfully ask, how are these higher frequency oscillations generated? We know that we can see them on the EEG, but they don't seem to be due to synaptic kinetics. So one hypothesis that's been proposed is that this is due to action potentials, the outputs of these neurons that I talked about previously. And so what we did is we went back to our models and characterized the EEG signals generated by the outputs. Previously, I was talking about the synaptic input. Now I'm going to be talking about the action potential output of these neurons. And essentially what we can do is we can model these same neurons. We can generate the electric fields. And you can see that generates these black curves here. The action potential is present. And then we can re-simulate these neurons with the action potential generated mechanisms taken out. Okay. So basically there are channels, as Anwar alluded to in the introduction, these nav channels that are important for the production of these action potentials. We take them out of the model, generate a completely silent neuron. And from this, we can see what the signal would look like purely from the spike. And if you were to just average over all these black lines and zoom in a lot, this is what a single action potential would look like on the EEG signal. Very, very small. And based on these, basically applying the same analysis for all the correlations that I talked about before, which I won't get into for time, we can then look at how the amplitude of these action potential generated EEG signals would look. And look at this across different frequencies and ask what fraction of an EEG rhythm is generated by action potentials versus synaptic currents. Now, as I mentioned, at a certain frequency, the synaptic currents are going to be attenuated so much that they can't generate EEG signals. And that's sort of this noise floor here. You can see that basically above 200 hertz, synaptic currents can't generate EEG signals. Depending on certain parameters that are unconstrained in the model, we can't really constrain where it is in the y-axis here, but we can look at what fraction of the brain rhythm is going to be contributed by action potentials, which I have colored in here in dark colors. At these lower frequencies, unsurprisingly, action potentials aren't contributing much. It's all synaptic activity, which is what people think. But above, as you get to higher frequencies where synaptic currents can't contribute anymore, you can actually generate very large brain rhythms purely from action potentials. In fact, above, say, 250 hertz here, you're either not generating any brain rhythm at all, any EEG oscillation at all, or you're generating an EEG oscillation from action potentials. So given the fact that we know 600 hertz oscillations exist on the EEG, they must be coming from action potentials. Synaptic currents would be too attenuated. Let's get through this for time. All that to say, we have a picture of the EEG spectra that looks like this. At these lower frequencies, we know that brain rhythms are generated by synaptic currents. At higher frequencies, they can't be generated by synaptic currents. We have some evidence, which I didn't really go into, that they're generated by action potentials. And then you also have this entire broadband component to the EEG signal, which is contributed by many, many different factors. And these factors are all operating in different ways, changing the spectrum in different ways. For example, you have the GABA receptor kinetics, you have the leak currents, AMP receptors, neural avalanches, muscle noise. And so this seems like, first of all, that the broadband spectral component EEG is very rich, and there's a lot to be learned from it by analyzing it. But it's also maybe a bit too rich to try to understand. Interestingly, our modeling framework actually provides a very nice organization for how these different factors influence EEG signals. Firstly, there are the neural dynamics. For example, changes in this network criticality that I was talking about, as well as some other things that I didn't get into. Changes in neural dynamics are going to interact with other changes in neural dynamics completely additively, because as I mentioned, electric fields sum linearly, and so if one part of the brain is now oscillating, the other part of the brain is now aperiodic. These changes are going to affect each other additively. Then there are the parameters that affect the core biophysics of EEG generation. These are the GABA receptor kinetics, the other things I haven't talked about, like the leak potential. And this will basically multiply all the power generated at a certain frequency. Changes in this need to be detrended because they're going to otherwise corrupt your EEG oscillation estimates. And then finally, there's all this external additive noise that's going to be true in any signal, such as the amplifier noise and muscle activity. So all that brings us to three main conclusions from the studies, the three simple conclusions that we can draw. The first one is that EEG spectral trend provides physiological information. It's not just noise to be discarded. It's not just an epiphenomenon of brain rhythms. It actually provides something interesting, some interesting information about what's happening in the brain. The second is that the EEG spectral trend reflects a multitude of mechanisms. There are those who might argue it's due to some universal critical state of the brain, but it's actually much richer than that. And there's many different things from the single neuron level to the network level that's going to change the spectral trend. I'd say that one of the big challenges moving forward is going to be inferring these changes from a purely data-driven point of view. We can simulate neurons and say, well, if you change this parameter, you're going to have this effect on the EEG signal, but we'd like to be able to go the other way around. Seeing, given two spectra, can we infer what's changing in the brain? And then finally, the EEG spectral trend requires rectification in certain cases. Now, our results caution against going around and detrending all EEG spectra for no reason, because sometimes these changes are completely innocent and they can be ignored. But in certain cases, specific when synaptic physiology is affected, then you might need to detrend. And this is going to be particularly important in any cases where you have some sort of pharmacological intervention. Almost all drugs alter brain activity by changing these sorts of parameters like channel kinetics. And so detrending might be particularly important in cases where you have individuals receiving pharmacological treatment for psychiatric or neurological disorders, for example, epilepsy, where it's known that they're receiving drugs that change potassium channels. So that brings me to this. This is the first EEG spectrum that was ever calculated. It was calculated in 1938, well before there were computers. And so to calculate the signal, they literally hooked up a physical system of projectors and wheels to physically transform the EEG spectrum being drawn out by a pencil into Fourier space. And upon seeing the very first EEG spectrum ever recorded, they concluded that inspection of such continuous spectrums should convince one of the inadvisability of referring to certain potentials as alpha, beta, delta waves, etc. Now, the success that we've had from characterizing all these waves in the last century suggests that they have a lot of utility, but I think there's a nugget of truth in this old quote. And I think the emerging field of studying aperiodic neural activity and aperiodic EEG kind of goes back to the roots that is shown in this quote. And maybe as a sort of naive observer, look at these signals and say, yeah, I see this peak, but what's this? And what does it mean? And what can we learn about the brain from it? And I think the answer to that question is a lot more than we're currently getting out of these EEG signals. So with that, I'd like to thank Anmar, my PhD thesis advisor, as well as Dr. Gilles, who collected this EEG data that I presented with the protocol kinetics. Thank you. I'd be happy to take questions.
And also go to the chat there because there will be some questions from the students a little bit here. Leon, go ahead.
Question, sorry, yeah.
Go ahead, okay.
So you talked about criticality
Mhm.
and you didn't say what happens on the other side of the criticality.
Mhm.
Can you comment on that? And anyway, I'll have a follow-up.
Yeah.
Maybe.
I mean, at least in the specific type of criticality we're looking at, the system is unstable above that point. And so, I mean, we can't really.
Is that because you don't include a refractory time?
Yeah, I mean, it's a very simple model of neural dynamics. But even if you had to have like very realistic models of excitatory neural populations and inhibitory neural populations, if the excitation gets too large, it can still just blow up and go to infinity.
But if, I mean, it was just curious that you talked about 0.98, 0.99, 0.9999,
Yeah.
whatever. But is the reason why, do you have a refractory time in your model, or it's just a cell can fire as frequently as it gets the input?
In this specific model, I mean, it's constrained by the fact that we have to do things discretely on the computer, but yeah, there's a refractory period in the sense that it can only fire once every 4 milliseconds.
But is that because your delta T is 4
Yes, exactly
milliseconds
right,
in your
yes.
model?
Yeah.
Okay, so,
But I think even if you implemented a hard refractory period, you can still get these, you're still going to get explosions in the overall activity.
but when people are studying avalanches,
Yeah.
they're kind of showing the synchronous firing of,
Mhm.
in lots of. I'm not sure how that would, there's a literature dealing with the avalanches and so forth. I'm not sure how that fits in with what you're doing.
Yeah, I mean, there's many different types of neural avalanches. I think a lot of the work on these neural avalanches are talking about sort of the physical size of them, right? So you can have like a 2-dimensional array of neurons, and the question is how many neurons are firing, and they tend to be spatially constrained. You can also talk about criticality from a purely, as I mentioned, these branching models, which can just be a single dimension, where you just have the average firing rate of the network, and this firing rate is dependent on the previous point times this branching number. So you have firing rate at time t is 0.98 times refractory period of the previous time. And in this sense, I think that there's slightly different types of avalanches that they're talking about.
Okay. Okay, thanks.
Questions online in the chat possibly, or if you want to just raise your hand and
Or just talk.
talk, yeah.
Yeah. Thanks. Yeah, thanks, Niklas. That was great. And I'm really happy that QLS can showcase the work of its alumni. Happy we can start doing that because the program is old enough, and it's great to see that we can start. Was that?
I said, we finally have alumni.
Exactly. And I'm really happy that we can start showcasing these our alumni with such a great talk. It was really, really interesting. I have many questions. Most of them are just questions for me to learn about that system. But in knowing a bit about criticality in other systems, if you can clarify something for me, I still take away from your talk that you were keeping the broadband spectrum and the rhythmic part as additive or as separate, right? Should we separate them? But it's hard for me to understand how a rhythmic force in a system would not mess with any process that would lead to criticality as a forcing that would just completely interact with it. So am I getting that wrong, or is it like an assumption that is reasonable given that system?
I mean, I think it's a very important question. I would start by saying, I think it is reasonable to think that certain parts of the brain are not entirely independent of one another, but that they can exhibit mostly independent dynamics from one another. For example, you could have the prefrontal cortex doing one thing and then visual cortex doing another thing. But I think it's actually a very interesting question. And there are some models where they try to capture this sort of criticality and oscillations in the same system. My problem with implementing some of these into my model is they already start to question the overall framework from analyzing EEG signals, because I think it kind of questions what a brain rhythm actually is. Because as soon as you get these models where there's kind of a continuous shift from sort of an chaotic regime to an oscillatory regime, at what point do you start calling it a brain rhythm? And I'm not quite sure that there's a good answer to that question.
Yeah. Okay. Thanks.
I see no other questions. So thank you, Niklas, for a wonderful presentation. And thank you all, everybody online, for joining us.